Okay, so on Thursday we were looking at electron transport chain. So let's go over mechanism, and then we will get into inhibitors and uncouplers of electron transport chain and brown adipose tissue. So when it comes to mechanism of ATP generation, so the most important thing is that your inner mitochondrial membrane should be strictly impermeable to protons. And your electron transport chain is located in the inner mitochondrial membrane. These are complexes which are embedded in the inner mitochondrial membrane. So we have 4 complexes, complex 1, 2, 3, 4, and they have the alternate names, which I told you to remember. And when it comes to complex 2, there are 3 of them. So there are 3 names for complex 2, right? So in complex 1, NADH is oxidized into NAD plus. As you see here, NADH plus enters into complex 1 and it is oxidized to NAD plus. That means H and H plus enters into complex 1 because NADH plus H plus, H and H, 2H, 2 protons, right, enters into complex 1. And similarly, complex 2, where FADH2 gets in and oxidized to FAD, that means 2 protons again enters into coenzyme Q. So now complex 1, after getting its protons from NADH plus, this complex 1 acts as a proton pump. You got to carefully look at this figure, and also compared to other figures, I would say this is, it will give a lot of information in one place. Earlier, I was just building up, and now you know the background information. So this figure you can use because the complex 1 other name is also given here. Complex 2, all you need to write down the complex 2 names. There are 3 of them, which is available in the previous figure. Then complex 3, BC1 complex, and then complex 4, cytochrome C oxidase. And so cytochrome C is there and coenzyme Q. Those are mobile complexes. Another thing here is complex 1, complex 3, and complex 4. See what is shown here. Complex 1, 3, and 4, they are pumping protons, as you see. Complex 1 is pumping 4 protons. Complex 3 is pumping 4 protons. And complex 4 is pumping 2 protons. That means out of all electron transport chain complexes, there are only 3 complexes which are acting as proton pumps. And those are 1, 3, and 4. That's important. That can be asked in the exam. Okay? These are proton pumps. Now, as they pump protons, so that means in order to pump protons, they need energy. You know, any proton pump, like sodium potassium pump. You know, sodium potassium pump? It needs energy, right? It breaks ATP into ADP. You need energy to pump something. Here also, to pump these protons, complex 1 needs energy, complex 3 needs energy, complex 4 also needs energy. How these pumps will get energy? The energy will come from oxidation of NADH plus into NAD plus. Oxidation, remember, it releases energy. And there is a flow of electrons. As you see, electrons will flow from complex 1 to coenzyme Q, from coenzyme Q to complex 3, complex 3 to cytochrome C to complex 4, and then finally to oxygen, from negative redox potential to positive redox potential. So, as these electrons will flow from the complex which has got relatively more negative towards the complex which has got relatively more positive potential, there will be loss of energy in that particular flow. The flow of electron, as they flow from one complex to another complex, they kind of go on releasing energy to these complexes, and that energy is used by the complex to pump protons. Okay, this is how protons are pumped across the membrane from the matrix side into the intermembrane space. See, this is the down here is the matrix as it is shown in the figure, and here is the intermembrane space, and protons are pumped using the energy that they get from flow of electron from negative potential to the positive potential. They kind of lose that energy. So that means when you oxidize NADH plus, you are oxidizing NADH plus into NAD plus. Let's consider that. Oxidation of NADH plus into NAD plus, the amount of energy that you get from that oxidation process will help in pumping 4 plus 4 plus 2, 10 protons from the matrix into the intermembrane space. So totally there will be pumping of 10 protons. Remember, there are 10 here, 4 plus 4 plus 2, 10 protons. So I'm going to write that for you. So NADH H plus oxidation into NAD plus and leads to pumping of 10 protons into intermembrane space. And that is what oxidation of NADH plus into NAD will do. So NADH plus, when it is oxidized to NAD plus, it helps in pumping of 10 protons into intermembrane space. Then you may be wondering from where it got 10 protons. NADH plus H plus is oxidized to NAD plus. How many protons you have? When NADH plus is oxidized to NAD plus, how many protons you got? NADH plus H plus. There are only 2 protons, right? NADH plus H plus oxidized to NAD plus. It is releasing 2 protons, right? Here, NADH plus into NAD plus. What is the difference between those two? Only 2 additional protons. But what I am saying, when NADH plus is oxidized to NAD plus, what did I write here? I wrote it helps in pumping 10 protons from the matrix into intermembrane space. So that means additional 8 protons which it did not have, from where it got? That's what my question is. From where it got that additional 8 protons which it did not have? You're getting my question? From here, from the matrix. It is simply getting those protons from the matrix. Remember, there are protons in the cytoplasm, protons in the matrix also. So these pumps, once they get energy from the flow of electrons, what they do, they get protons from the matrix and pump it into intermembrane space. Okay. So that's the point you got to remember in case if we get confused later on. So although there is only 2 protons in the oxidation of NADH plus into NAD plus, but the amount of energy that it gets from the oxidation of NADH plus into NAD plus, that energy is sufficient to pump 10 protons. Okay. Now, if you carefully observe this figure, complex 1 is pumping 4 protons. Complex 3 is pumping 4 protons. Whereas look at complex 4. It is pumping 2 protons. Why not 4? If that is what is the question. The answer is, by the time the flow of electron go from complex 1 to coenzyme Q, then complex 3, cytochrome C, it is going on losing energy. By the time it reaches complex 4, the amount of energy that is left, it can only sufficient to pump 2 protons, not more than that. You're getting the point. Not really super high yield, but you should have an idea why it is pumping 2 protons. Okay. Anyway, so oxidation of NADH plus into NAD plus helping in pumping of 10 protons. I want you to know that because we are going to use that information for ATP calculation. Yes. Correct. 4 plus 4 plus 2, 10. So every NADH plus being oxidized, it will pump 10 protons from the matrix into intermembrane space. Remember that. And that 10 come from 4 plus 4 plus 2. Okay. Now, let's look at oxidation of FADH2. Let's focus on FADH2 now. I have written FADH2 here. If you want, you can write it down so that figure becomes like kind of little bit of complete. FADH2, as you know, it enters into complex 2. I have written complex 2 down here. There are 3 complex 2s. You know that. So FADH to enter and oxidize to FAD plus, and that will be given to coenzyme Q, and coenzyme Q will carry on. So that means FADH2 don't enter into complex one. That's the thing you got to remember. FADH2 never enters into complex one. It's only complex 2. It is oxidized in complex 2. So it is going to give its protons to coenzyme Q, which becomes coenzyme QH2. Okay. So then what happens? The energy that it gets from oxidation of FADH2 into FAD, it is since entering into coenzyme Q and later, right? So the pump that is available immediately next is complex 3, not complex 1, because it has bypassed complex 1. It is entering into complex 2, coenzyme Q, and then complex 3 is the pump now. That means it is pumping 4 protons, and the next complex that is acting as a pump is complex 4, which is pumping 2 protons. That means when your FADH2 is oxidized to FAD plus, or FAD, not FAD plus, FADH2 oxidized to FAD. So it helps in pumping how many protons now? 6 protons into intermembrane space. All of you clear with this? Why 6? Why FADH2 oxidation gives you means helps in pumping 6 protons instead of 10? Because it bypasses complex 1 basically, right? It enters into coenzyme Q and then complex 3. So it starts from complex 3 there. Pumping starts from complex 3. So that is why, so that will be 4 plus 2. That will be 6. Is it clear, all of you? Okay. So these numbers will help us to calculate ATPs later. So for NADH plus, it is 10. For FADH2, the amount of energy that is there in that molecule is helping in pumping only 6. So 10 and 6. Okay. Those are the numbers. Now, as this pumping continues, as more and more NADH oxidized, more and more FADH2 oxidized, as the pumping of protons from the matrix into intermembrane space continues, so what will happen to the proton concentration in the intermembrane space? It will go up, right? Continuously you are continuously pumping water or something into the space. What happens? There will be a lot of water building up there, right? Same thing here. There will be more protons kind of building up in the intermembrane space. There will be a lot of protons building up in the intermembrane space. So proton gradient in the intermembrane space increases. There will be more protons in the intermembrane space compared to the matrix eventually. Okay. As this more and more protons are building up in the intermembrane space, so the proton gradient will be created where there will be more protons in the intermembrane space, less protons in the matrix. So now what happens? Because there is a lot of proton concentration in the intermembrane space, it will build a potential, and that is called as electrochemical potential. And this electrochemical potential, what it does, it has got energy. Because a lot of protons building in one space creates energy. And that is called electrochemical potential. And that electrochemical potential, what it does, eventually, these protons, they start to come back into the matrix. And there is only one way for them to come back into the matrix, and that is through this complex called ATP synthase. That is the only one way. Because complex 1, 3, and 4, they are pumping from matrix into the intermembrane space. Whereas ATP synthase is able to allow protons to get back into the matrix. Okay. So, and that requires only, there will be a lot of electrochemical potential is needed, more protons should be there, gradient has to be there, then only it will allow. So that is what is shown here. Now, n number of protons, n means it can be any number. N number of protons start to enter into the matrix. Say they flow down like this, H plus through ATP synthase. That's your complex, and this ATP synthase, it is sometimes referred as complex 5. Complex number 5. is ATP synthase. Write it down in case. ATP synthase is also referred as complex number 5. The role of ATP synthase is it will allow flow back of protons from the intermembrane space back into the matrix. Okay. These protons are coming from the intermembrane space where there is a electrochemical potential created and they are falling back into the matrix side. So there is a flow of those protons there. As these protons are flowing back into the matrix through this ATP synthase, there is energy in that flow again. Because that's an electrochemical potential, there is an energy which is stored in that. As they flow back, the energy that they carry, it is used by ATP synthase to combine ADP with PI to make ATP. And that is what is shown down here. As you see, in the matrix, as there is a projection of ATP synthase into the matrix, in that projection, we are going to see what is the subunit and all that in the next slide. So there is ADP which is combining with PI to make ATP. So ATP is generated because of flow of protons into the matrix from the intermembrane space. And this is how you are causing phosphorylation of ADP with PI to make ATP. See, that's a phosphorylation process. And what we have done to NADH and FADH, NADH plus H plus and FADH2, what happened to them? They are being oxidized. As NADH and FADH are oxidized, you are causing phosphorylation of ADP with PI to make ATP. That is why the electron transport chain complex is called as oxidative phosphorylation. So that means oxidation is necessary for the phosphorylation process to happen. These are coupled processes. And what if you uncouple them? What if you block this oxidation process? What will happen to the phosphorylation? It doesn't go on. That means ATP synthesis will decrease, right? So what makes them to be coupled? The coupling process is possible only if your inner mitochondrial membrane is strictly impermeable to the flow of protons other than through the pumps. They should not simply go in and out. The traffic should not be free. It has to go through pumps. Okay. So the pumps here, complex 1, 3, and 4, they are unidirectionally from the matrix into the intermembrane space, whereas ATP synthase is also unidirectional but in opposite direction from intermembrane space into the matrix. Only they have to go through these pumps, these proteins, right? So what I will do now is I'm going to create a pore in the inner mitochondrial membrane. Let's consider that I have used a chemical or a molecule or something like that, and I'm going to create a pore in the inner mitochondrial membrane. So I'm going to insert a molecule which is just allowing a flow of protons from the matrix into from the intermembrane space into the matrix. So I'm going to bring these protons which are kind of building up there in the intermembrane space. I'm allowing them to run down here. And that I will call it as, this is an ionophore. Ionophore in the sense they are lipid soluble molecules, and I'm kind of putting them or inserting them in the inner mitochondrial membrane. And what they do, they just allow free flow of protons. So what happens to your electrochemical potential if I am allowing this free flow of protons? Every time these protons are pumped from complex 1, 3, and 4, they are kind of coming back into the matrix side through this gap or the pore that I have created. What happens to the electrochemical potential? Does it build up? It doesn't. Electrochemical potential doesn't build up. Why? Because you are leaking the protons. You're leaking the protons. When you leak the protons, so you are not able to build chemical potential. That means what happens to the ATP synthase function? will decrease because ATP synthase, it works only when there is a sufficient electrochemical potential built. Now you are not allowing it. So ATP synthase simply fails. That means phosphorylation simply fails. You're not keeping up phosphorylation with oxidation. And that's when you uncouple two things. There is an uncoupling process. There is electron, means oxidation going on. There is no issue with oxidation. NADH oxidize, FADH oxidize, your complexes are pumping protons, but simply those protons are returning back through the alternate channel, that is the pore. So phosphorylation is not keeping up with oxidation process. This is an uncoupling process. And who has uncoupled this process? This molecule which is inserted in the inner mitochondrial membrane. And that molecule which is there in the inner, which is inserted into the inner mitochondrial membrane, which is uncoupling oxidation from phosphorylation, you simply call that molecule as uncoupler because it is uncoupling. This is an uncoupler. It uncouples oxidation from phosphorylation. And that's how uncouplers will work. Okay. So uncouplers, what they do to your inner mitochondrial membrane? They simply allow free flow of protons. Is it not? Uncouplers simply allow free flow of protons through the inner mitochondrial membrane. So that's why we won't be able to build the gradient. So we'll come to know what are the different types of uncouplers that we need to remember and all that. Okay. So before I move on, so the protons that are coming back through this uncoupler instead of ATP synthase, although there is no that much electrochemical potential is not built up, but still every proton has some amount of energy, little amount of energy. It's still having. Whenever the proton flows, it does have some amount of energy. And they are going through this uncoupler instead of ATP synthase. So ATP synthase, if it went through ATP synthase, we would have got some product that is ATP. Whereas here, it's kind of, we won't be able to tap that energy. So where that energy goes? The small little energy that is coming from proton flow through the uncoupler, what happens to that energy? It's simply released as heat because you're not tapping it systematically. So it is simply released as heat. So when flow of protons occurs through uncoupler, there will be release of heat. I'm going to write that here. You please add it. There will be release of heat. So whenever there is a mechanism of uncoupler, means uncoupler is doing its function for whatever the reason, physiologically, chemically, whatever. So there will be release of heat. And that heat, it can bring up your body temperature. And that's why when there is a toxic effect of uncoupler, or whenever there is an uncoupler mechanism going on, especially with the chemicals and drugs, what will happen to your body temperature? Will shoot up. Remember that. That's how you are going to identify, oh, there is an uncoupler going on here. In the case stem, you are going to see that. Yes. I'll come to that point. Any increase in body temperature is fever. Okay, uncoupler will also lead to fever, but it's not infectious, right? So the point that you should remember is the mechanism, the way how uncoupler is working, what it does to your body temperature, it will increase. Remember that. Increase in body temperature, it should indicate there is an uncoupling mechanism going on. Okay, again, of course, you got to connect with the case stem, how it is built and all that. But in the case stem, if there is a chemical and if there is an increase in the body temperature, and if it is something, the choices are something related with the electron transport chain, you should think about uncoupler. You got the point? Okay, so this is how the electron transport chain is kind of built up and how it is organized and how the protons are moving and how they are pumped into the intermembrane space, how they come back through ATP synthase to cause phosphorylation, and how uncoupler can interfere with this process. Thereby, there is a uncoupling process. Okay, so this is what is all about uncouplers of electron transport, means introduction to uncoupler and overall mechanism of electron transport chain. Before I move on, I'm sorry about this. What about inhibitor? Let's consider that. Let's forget about uncoupler. Let's consider you have an inhibitor of electron transport chain. Let's inhibit complex 1. All of you. Let's inhibit complex 1. You just block complex 1. Let's see what happens. You block complex 1. Just for the sake of understanding, I'm going to put this mark here. Complex 1 blocked. There is a complex 1 inhibitor. It is blocking complex 1. Okay. What happens to your oxidation of NADH into NAD plus? It's not going to oxidize because your complex 1 is not working. So that means NADH will build up. NAD will be less, more NADH. Is it not? They're not oxidizing, so they're just building up. That's one thing. Then what happens to pumping of complex 1? Proton pumping up complex 1, 4 protons. Generally, it is to pump, right? What happens to that? It's not going to pump because you're not getting protons there. Okay. So if it is not pumping, so that means these 4 protons entry into intermembrane space, you don't get it. Does this mean FADH2 is not oxidized? Is it possible for FADH to be oxidized into FAD and help in pumping of protons from complex 3 and 4? Yeah, why not? If I fall sick, right, and not able to come to class tomorrow, Dr. Sebastian will come and teach. Is it not? There is a other way to do it. Here also, if complex 1 is blocked, we have complex 2, where FADH2 can get in and being oxidized. Only thing is, it can pump only 6 protons. That means it will take more time to build electrochemical potential. That means the amount of ATPs that you get on any given point in time slightly reduced, but not stopped. Is it not? Is it not? FADH2 is there still, it can be oxidized. Only thing is, how much FADH2 your metabolism produces, right? So, because really in a relative sense, there are a lot of NADH, there are so many metabolic pathways where NADH is produced compared to FADH2. That is why ATP synthesis can still go on, but at a reduced rate. It doesn't stop. That's the point. So, inhibiting complex 1, you are inhibiting oxidation of only NADH, but not FADH2. Okay? Okay. Now, what I will do is, I am going to inhibit complex 3. I am going to use a inhibitor of complex 3. Complex 3 inhibitor. Now, what happens to oxidation of NADH and oxidation of FADH2? So, it will affect both, right? Because complex 3 is necessary for both complex 2 and complex 1. So, now that's a common path that everything has to go through that complex 3, right? And you are blocking it. Highway blocked, that's it. This road here, St. Kitts, blocked. How do you go to Royal? That's the only road. From where you go, you have to get onto the hill. You can go by walking, no car. I'm just saying, that's a highway and you are blocking. Complex 3 is necessary for both complex 1 and complex 2 and you are blocked. That means oxidation of FADH and oxidation of NADH both will be down. So, what will happen to ATP synthesis? Totally down. And so, what happens to the NADH levels and FADH2 levels? Will build up. NADH and FADH, when they are not oxidized, they will go up, they will build up because you are blocking complex 3. Like this, the inhibitors of electron transport chain will work based on where you are inhibiting it. Similar mechanism if you inhibit complex 4. Say if you inhibit complex 4. Okay. If you inhibit complex 4, same thing. Again, FADH2, NADH2 not oxidized because that's the common path for both of them. So, like this, inhibitors and uncouplers will work. So in the presence of inhibitor, so there will be overall decrease in ATP synthesis. Even in uncoupler also, there is overall decrease in ATP synthesis. Okay. Now, let's come back to uncoupler. Uncoupler of electron transport chain, the last concept. When uncoupler is working, so there is a oxidation of NADH and FADH2. There is pumping of protons. There is no problem with that. Only thing is these protons are freely flowing back into the matrix. And that's why there is no enough protons to go through ATP synthase. That's why significantly the phosphorylation of ADP with pi to ATP decreases. That means what will happen to ATP concentration in the matrix? It decreases. ATP concentration decreases. What happened to ADP concentration? ADP increases. Okay, let's come to that. So ADP concentration increases, is it not? Because you are using uncoupler, you are separating oxidation from phosphorylation, uncoupling. The phosphorylation not going on means your ADP is not phosphorylated with pi. So ADP level increases, ATP level go down, ADP increases. So if you want to write ATP down, which is the opposite one, okay, ADP level increase. Lack of ATP, buildup of ADP. What is the energy status of your cell? Down. Energy status down, your ATP levels down, and cell is in kind of stress. And you don't have dollars, what you do? You came to send kids. No dollars left. Just 4 more weeks to go and no dollars. Is it not? Be careful with your finances. If you have limited resource, you got to play within that. Can't just go and do party and no dollars and fast for 4 weeks. No. You got to be disciplined and careful when you spend. Every dollar counts. You know, life lesson. Sometimes we learn it by hard way. I did that. Okay. So be careful with your money. Limited amount. Got to be careful, clever, and use it wisely. Okay, I don't have time to tell my story. I will tell some other time. Okay, because I have to save time for your mock exam. It is there in my mind. Mock exam, I have to save 1 hour. Okay, and also I have a couple of questions for eWorld review questions. So, what I'm trying to say here is, when ADP increases, lack of ATP, so cell is kind of stressed. What it will do? How to generate ATPs? How to generate ATPs? How we were the cell was generating ATPs. It was oxidizing NADH into NAD plus, oxidizing FADH2 into FAD plus, pumping protons and creating chemical gradient, and then allowing those protons to come back and make ATP. That's what it was doing. That means your cell has to first make this NADH and FADH2, right? You should have FADH2 and NADH, then only you can oxidize them. How to get NADH and FADH? Metabolism. You got to run metabolic pathways. Okay. So the metabolic pathways, I'll just write one metabolic pathway, which is more common one that we all study. That is oxidation of glucose. Okay. So glucose is oxidized to pyruvate. We're going to study this. And pyruvate is oxidized into acetyl-CoA. And acetyl-CoA undergoes TCA cycle, as you all know, TCA cycle. And in the TCA cycle, you're going to get NADH made, NADH, and also you're going to get FADH2. That's what gives you NADH plus H plus, of course, and HAD and FADH2. NADH plus H plus and FADH2 you get from metabolism. So overall, I'll write it as metabolism. What kind of metabolism? Is it anabolism or catabolism? How do you make NADH and FADH? It is through catabolism. You are oxidizing. So one of the catabolic pathway, oxidation pathway, is glycolysis. Glucose is being oxidized. So glucose to pyruvate, pyruvate to acetyl-CoA, and acetyl-CoA TCA cycle, and you get NADH and FADH. So remember, both from oxidation of pyruvate to acetyl-CoA, you're going to get NADH here, and acetyl-CoA going into TCA cycle also, I have written, you're going to get NADH and FADH. Okay. You are looking at uncoupler, which is uncoupling oxidation from phosphorylation. ATP levels are decreased. That means ADP level increased. And cell wants to make ATP. And if the cell wants to make ATP, what it has to do to oxidation means the catabolic pathways, oxidation pathways. It has to increase oxidation pathway, right? You want to make ATPs. That means cell has to increase its oxidation pathways, trying to get more and more NADH and more FADH, thinking that these NADH and FADH will go into complex 1 and complex 2 and pump more and more protons, trying to build electrochemical gradient, thereby you get some ATPs. That's the idea. That's what cell wants to do, right? So what cell will do? Cell will, this increase in ADP, this increase in ADP here, what it does is going to promote TCA cycle. It's going to keep your TCA cycle super high. It is going to promote that. It's going to stimulate TCA cycle, right? And thereby it is trying to make more and more NADH and FADH2. It will. This increased ADP will regulate positive allosteric modulator on a TCA cycle, thereby make more NADH and FADH2. Even it acts on the conversion of pyruvate to acetyl-CoA, oxidation of pyruvate. So it may create NADH and FADH. What happens to them? They will enter into complex 1, complex 2, and then pump protons, help to pump protons and trying to create a gradient. Will that create a gradient when there is an uncoupler? Does it do it? Will it be successful? When there is an uncoupler down here, which is allowing free flow of protons, but still your ADP, increase in ADP is stimulating TCA cycle, pyruvate oxidation, making NADH, FADH here, and trying to pump protons, trying to build the gradient, electrochemical potential, but these protons are continue to leak. Protons are continue to leak, right? So will it be successful in creating gradient? Partly. What we will do is, we will take a water container. Let's consider, use bottle, Alex, yours, okay, don't mind taking this. Okay, this is your water can, right? What I will do, I will put a hole here and put a tap. Consider it's a big barrel and there's a tap. Okay, and water from the hose pipe, it is entering into this particular container on top. Water is flowing in, right? And unless I close the cap here, means the tap should be closed, right? If you want to fill the bottle, you have to close the tap, right? And that's how electron transport chain will work. It has to close the tap. That means there should not be uncouplers. Now what I'll do, I'll insert uncoupler. I'll open a pore here and allow it to flow. I'm putting a pore here. Allow it to flow, and on top I am putting water. Also that hose pipe is filling this bottle, but here I am allowing it to flow. Will you be successful in filling this bottle? Even if you increase the pressure up there, you may try to fill a little bit up, but never full. Right? And that's what happens with the uncouplers. Your cell is trying to increase metabolic pathways, increase catabolism. So, trying to increase NADH plus in the hope that these will be oxidized and able to, build sufficient gradient to get ATPs, but it won't be 100% successful. So, your free flow of protons will continue to happen, and that means ATP levels continue to be down, ADP levels are continue to be up, and it keeps on stimulating this Oxidation pathways continuously. That means person has to take more and more food, but not able to generate sufficient ATPs. And that creates high basal metabolic rate in that person. Because you are increasing the metabolism, right? When you are increasing the metabolism, your BMR increases, basal metabolic rate increases. And because of that increase in metabolism itself, these oxidation pathways also release energy as heat. Remember, only 48% is tapped as ATPs, 52% is released as heat when you're oxidizing glucose. So that 52% which is going at higher rate, plus leaking of protons through uncoupler, which is also releasing heat. What will happen to the overall body temperature of this person? Goes up, right? So that's why when uncoupler is working, so there will be increase in body temperature. And what will happen to the basal metabolic rate? What will happen to the basal metabolic rate? BMR in the presence of uncoupler will shoot up. BMR increases, body mass, body temperature increases. Okay, last concept. So when electron transport chain is continuously running, in fact, it runs faster than before, trying to build the gradient, because your basal metabolic rate increases, there will be NADH coming in, FADH2 coming in, your electron transport chain is overworking, trying to build the gradient, right? But it is not being successful, right? So in the last complex of electron transport chain, that is complex 4, see what is going on. What happens to the protons? Who is the final acceptor of your proton? More positive redox potential. Which molecule has got highest positive redox potential in the electron transport chain? And that is oxygen, right? Oxygen is the final acceptor of protons. So when you are keeping your electron transport chain super high in hoping to build the gradient, what happened to the oxygen consumption? Will also go up. Remember, I told you whenever there is a rise in the means increase in electron transport chain, there is a need for oxygen. That means you got to continuously supply oxygen for this increased electron transport chain in the presence of uncoupler. So the consumption of oxygen increases. And increase in oxygen consumption means what? Your respiratory rate increases. So that's why in the presence of uncoupler, what uncouplers will do? Uncouplers increase temperature, increase BMR, basal metabolic rate, increase oxygen consumption, and thereby increase in respiratory rate. I'll write it as RR. You write it as respiratory rate. Don't write RR, because later you may wonder what is RR, right? A respiratory rate. There is increase in respiratory. This is what, and what happens to the ATP levels? If you want to write down ATP will decrease. So I'll write down here up there. Decrease in ATP. That is common both for inhibitors and uncouplers. ATP, yes. BMR, basal metabolic rate. BMR. Basal metabolic rate increases because you are keeping your catabolic pathways high. You are oxidizing glucose, fatty acids, all that. So this is what is the effect of uncouplers. So this is the information that will be given in your STEM indirectly. So if you see the person has got higher body temperature, there is some chemical working on his body, there is increase in higher body temperature, and there is the respiratory rate of this person is increased. You know, it just increased respiration, they're trying to get more and more oxygen, right? So, and there is a decrease in ATP levels. It is indicative of uncouplers. I'll give you some examples later, questions. All right. So that's about the inhibitors and uncouplers and overall mechanism of ATP generation. So I'll just move on now. This is all just continuation of what I have explained before. Okay, here is your ATP synthase. This is the structure of ATP synthase. Just a couple of points. If you look at the ATP synthase structure, it is also referred as F0, F1 ATPase. because it has got F0 and F1 subunit. F0 subunit, it is there embedded in the inner mitochondrial membrane, this part here, that is F0. So one which is allowing protons in, that's F0. This is F0 subunit. And then the F1 subunit is this one, which is the stock, which is projecting into the matrix. This figure is drawn upside down. It would have been really useful if they would have flipped this figure. Is it not? If they would have flipped it, then it would have matched with our previous figure. Look at this one. This is the right way of writing ATP synthesis. I tried to find the one that is flipped. Unfortunately, when I searched it, the resources, I didn't get. Okay, I'll keep searching. Anyone, if you get good figure, flipped one, send it to me. Okay, so but don't waste your time on that. In case if you get here and there, whatever, you just send it to me. Okay, don't spend time only on that, which is not really that important. As long as you know the stock is down. Okay, and next point that I want you to know is where exactly phosphorylation happens in this ATP synthase. That point you need to know in case if it is asked. I have not asked so far. And I did not see questions on that in any source. In case, just to be on safer side, where exactly, which subunit of F1, here it is, F1, it is shown here. As you see, ADP combines with PI, and which subunit they are shown here within that F1, which is the subunit there? Beta subunit. Beta subunit is a catalytic subunit in ATP synthase. That's where phosphorylation happens. Okay, and that is the point. So beta subunit catalyzes phosphorylation of ADP with PI. I have written here. And the last point, when this, and look at how many beta subunits are there in that stock, which is projecting into the matrix. How many beta subunits are there? 1, 2, 3, and each beta subunit can make 1 ATP. ADP with PI to make ATP. That means as the flow of protons occurs through F0, as you see here, here is the F0, as the flow of protons happens, look at the arrows, the flattened arrows, what it is shown. It moves, it turns. As the proton enters, it takes a little turn. Another proton enters, little turn. Another proton enters, little turn. Like that. Every time when proton come from intermembrane space into the matrix through this F0, it takes a little turn, right? As it takes a little turn, for the complete turn to happen, like 100%, like 360 degree, it takes 12 protons for one complete turn of F0 subunit. That's my statement, what I have written there. It takes 12 protons for one complete turn of your F0 subunit, making total of 3 ATPs. So one complete turn, 12 protons enter, and 3 ATPs you're going to make. Now, what I was trying to say was, how many protons do you need to make 1 ATP? That's what I was trying to say, but I got confused myself. So it takes 4 protons to make 1 ATP. Is it not? 12 protons making 3 ATP means entry of 4 protons making 1 ATP. Okay, that's the point. Now, with this information, use earlier what we have written, how many protons, oxidation of NADH plus into NAD plus helps in pumping how many protons from the matrix into intermembrane space? NADH plus oxidation, 10 protons, right? So those 10 protons which are pumped into the intermembrane space, when they return back into the matrix through ATP synthase, so how many ATPs it will be effectively contributing to? 2.5. Is it not? Because for every 4 protons you make 1 ATP, your NADH plus H plus helping in pumping 10 protons. So how many ATPs? So 4 protons, it is 1, another 4 protons, another 1, and then 2 protons, 0.5. Then you may ask, where is 0.5 ATP? Either it is 1 or 0. No 0.5 ATP, right? So 0.5 in the sense the energy. ATP is it just indicates equivalent of energy. 1 ATP is minus 7.3 kilocalories of energy. So 0.5 ATP means minus 7.3 divided by 2. That much energy it is worth, right? So that's why 10 protons pumping from NADH plus will help to make 2.5 ATP. That's the number, 0.5, 2.5. Similarly, do that math for FADH2, which is helping in pumping 6 protons, and when they return back, it will be 1.5. 4 plus 2. 4 is 1, 0.5 is from 2 protons. So that's the calculation there. I hope you got this, right? So NADH is 2.5, FADH2 is 1.5. So that's all for now. Let's do these questions. We'll have a break. When you come back, we will go into specific examples for inhibitors, specific examples for uncouplers, bone adipose tissue. Okay, let's do this question. So I have introduced this when I started electron transport chain. This stem, it's the same stem. So now you all know what is the vitamin that is deficient, right? Diarrhea, dermatitis, dementia. So the vitamin that is deficient is niacin, vitamin B3. And niacin is contributing to the coenzyme for which complex in the electron transport chain? It is complex 1. Remember, complex 1. NADH plus enters into complex 1 to oxidize into NAD plus. So deficiency of niacin will lead to deficiency of NADH plus H plus or NADs overall. So complex 1 is affected. That's why your answer should be complex 1.